topic that we were supposed to have done yesterday afternoon. And then we shall also look at glucose metabolism, and we shall also look at adrenal medulla. So depending on the time that we have, we'll try and do as much as we can. And also we also have another lecture which was supposed to be today for hormone metabolism and basic nutrition, but I thought it's important that we start with calcium and glucose metabolism and diabetes because they are more common. It's more common. So are you able to see the screen?
Yes.
You can see. Okay, great, great. Okay. So the purpose of this lecture is for you to be able to, oops, for you to be able to understand how calcium works in the body and the requirements for calcium. You shall be getting a lot of patients who require calcium, and all the patients require calcium from children to adults, even if you're going to do surgery. And in all aspects of medicine, calcium is important. Whether you're in ICU, whether you're in the community doing preventive, calcium is important. So calcium is one of the minerals that is important for normal growth and maintenance of the body. We have other hormones that also play a role in growth and development, and we shall also mention this. But specifically, calcium is important for the calcification of bone. It's also important for blood coagulation. It's a factor 4 among the clotting cascade. It's also part of the neuromuscular, it stabilizes the neuromuscular system to ensure that the irritability is reduced. It's also important in maintaining acid-base equilibrium. If you've done renal physiology, or if you're going to do renal physiology, you realize to be important. Also in maintaining fluid balance and osmotic regulation. So usually we say that calcium is a macronutrient because its daily requirement exceeds 100 milligrams. And if any other mineral has a daily requirement that is less than 100 milligrams, then it is classified as a micronutrient. So calcium is classified as a macronutrient. Calcium is the most abundant mineral in the human body because we have a skeletal system, and most of the calcium that we have in the body is bound to bone. And therefore, we find that the total amount of calcium we have in the body is easy to remember. It's about 1 to 1.5 kilograms. And 99% of this is bound to bone. And therefore, it's also bound together with phosphate. So usually you cannot be able to separate calcium from phosphate. When you break down calcium from bone, you also will lose phosphate from bone. So the levels will actually change. So what remains is a small percent in soft tissue and about 1% in the extracellular fluid. And this is why it has to be under tight control. So we have dietary sources of calcium, and the milk is one of them, which is a good source. But you also have eggs, fish, cheese, beans, lentils, and cabbages, and vegetables. So you see we have a wide variety of sources of calcium. What is the daily requirement? Usually adults need about 500 milligrams in a day, and children 1,200 in a day. Pregnant women and lactating mothers basically need slightly more than children, 1,500 milligrams per day. So by the time you're at the age of 50, there's a tendency for osteoporosis, and this is normally prevented by taking in more calcium than you should. So an adult should take in 500 milligrams per day, but in average then we need 3 times as much, which is 1,500 milligrams per day, plus vitamin D, which is about 20 micrograms per day. So we've said the body distribution of calcium is 99% in the bones, and usually it exists either as a calcium or a phosphate combination. And we've said we have 0.5% in soft tissue and 0.1 in extracellular fluid. So let's talk about plasma calcium. We basically have 3 types of calcium in plasma, and this is based on what I've discussed. It can be ionized, it can be free, it can also be unbound. So in blood, we have 50% of the calcium being free and being metabolically active. And we remember we said it's required for nerve function, maintain membrane permeability, contraction of muscle, and also hormone secretion. So among the proportion of bound calcium is 40% of the calcium is bound to protein, normally albumin. And this is why when you're requesting for a plasma calcium for a patient in the clinic or in the hospital, you should always request for an albumin level, because you have to adjust for albumin levels if the calcium is too high, or especially if the calcium is too high or too low, because in the situation where we don't have albumin, like malnutrition or loss of albumin due to liver disease or kidney disease, then you may overestimate the amount of calcium that is in the blood, in the body, because most of it will be in circulation. It will not be bound to most of it will be in the circulation, so it will not be bound to. Trying to close. It will not be bound to the albumin, so we have to adjust for that. So always remember, whenever you're requesting for calcium within when you're trying to estimate the levels of calcium in a patient, you must always request for a liver function test or albumin to be able to identify, to adjust for that calcium. So calcium has two forms, which is diffusible from both blood and the tissues, and we have the complex calcium, and 10% of the plasma calcium is normally complexed with anions such as bicarbonate. So you have calcium bicarbonate, calcium phosphate, calcium lactate, calcium citrate, and all these three forms of calcium in plasma remain in equilibrium with each other. So what are the normal levels of calcium? So we are looking at normal levels, and this you must know as in physiology. So it's 2.4 plus or minus 0.2. So you're looking at 2.2 to 2.6 millimoles per liter. You can also have it in milligrams per deciliter, but usually we prefer to use millimoles per liter. How is calcium absorbed? So the mechanism of calcium absorption has been described, and it's normally taken in the diet. We've talked about the different foods that have calcium. So not all the calcium that is taken is absorbed. So usually we have about 40%, only 40% is absorbed, and the absorption will take place in the second part of the duodenum. And of course, the absorption is against a concentration gradient, and it is energy dependent. Usually, calcium requires a carrier protein to help which is helped by the calcium dependent ATPase. 400 milligrams are usually excreted in stool, so sometimes you want to measure fecal calcium, and 100 milligrams is excreted in urine. We have two mechanisms of calcium absorption. It is simple diffusion or active transport. Simple diffusion, I think, is easy to understand because we said calcium is normally absorbed across a concentration gradient. Active transport, we said also calcium uptake is absorption is an energy dependent process, and we talked about the calcium dependent ATPase. And so this process requires energy to be able to move, and also requires a calcium pump. So both of these processes will require calcitriol, which is 125 dihydrocholecalciferol, which regulates the synthesis of calcium binding proteins and transport. So what factors will increase absorption of calcium? We have several factors listed here, and these are important to note because when you think about how to improve absorption of calcium in a patient, these may be some of the approaches that can be used or suggestions that the patient can actually be able to use to be able to increase the amount of calcium that they're taking in. One of them is vitamin D or calcitriol, and what it does is that it induces the synthesis of calbindin, and calbindin is a carrier protein in the intestinal epithelial cells, and this facilitates the absorption of calcium. We also have parathyroid hormone, which increases calcium transport from the intestinal cells by enhancing the enzyme 1-alpha hydroxylase. And we know parathyroid hormone is secreted from the parathyroid glands. The other factors that we also need to think about has to do with pH. pH is important because acidity will favor calcium absorption, and therefore we find that calcium salts, particularly calcium phosphate and carbonates, are quite soluble in acidic solutions. So therefore, it means that an alkaline medium will lower the absorption of calcium due to the formation of insoluble tricalcium phosphate. Also, a high protein diet, and I think these are some of the things that you'd also want to consider if a patient requires to absorb more calcium. A high protein diet will favor calcium absorption. So if you get a patient who has osteoporosis, or a pregnant mother or a lactating mother, or even a child, you'd want to give them a high protein diet so that you can be able to increase calcium absorption. So the reason for this is that if the protein content of the diet is low, only 5% of the calcium is absorbed. Remember, we said 40% of the calcium is absorbed, but if you have low proteins, then of course you see only 45% will be absorbed. So proteins are important in calcium absorption. The other factors that we have are amino acids, and amino acids basically will include lysine and arginine, which are important because they increase calcium absorption. Basically, how amino acids do it is that they increase the solubility of the calcium salts, and this, of course, will favor absorption. We also have sugars and organic acids, which will also are very important in producing microbial fermentation of sugars in the gut, and this increases solubility of calcium salts and increases the absorption. Citric acid, this is oranges and any kind of fruits that have vitamin C. Citric acid will also increase decrease absorption of calcium, and this one we said it's phytates, which we have in cereals, phytic acid, such as um, inositol hexaphosphate, and also insoluble calcium salts, and which we also decrease absorption. So think about cereals reducing the amount of calcium that we take, and it's been proposed that some of the porridges that some children take, which have a lot of cereals, may also put them at risk of getting rickets because of this. We also have Oxalates, which are from the green leafy vegetables, and they're important because they form insoluble calcium oxalates. Fibers also will interfere with absorption when they're in excess, and therefore, when you're thinking about a high fiber diet, always think about the possibility of calcium malabsorption. Malabsorption syndromes are usually very common cause of calcium deficiency because the insoluble calcium is bound to the fatty acids, and that then will cause will cause malabsorption of calcium. Glucocorticoids will also reduce the amount of calcium we have in the body by reducing intestinal transport. Phosphate, if you have a very high phosphate content, it will quickly bind to the calcium and precipitate it, and therefore reduce absorption. Magnesium also in high levels will reduce absorption, and there's a ratio of for every two molecules of calcium bound to magnesium, one of phosphate is bound to it also. In terms of biochemistry, calcium is important in the development of bone. Bone is actually a mineralized connective tissue, and it forms a reservoir of calcium. We've said it's about 1 kilogram, and it basically means that the bulk of it is in bone and teeth, and osteoblasts are important in forming bone, while osteocytes are important in demineralization of bone. Where does calcium act? Calcium acts in muscle by mediating its excitation contraction coupling and also interacting with troponin C to trigger muscle contraction. It activates ATPase and increases the action of actin and myosin and facilitates excitation contraction coupling. Calcium is also important because it will decrease neuromuscular irritability, and also we normally see this when we have tetany in calcium deficiency. Nerve conduction is also mediated by calcium because calcium is necessary in the transmission of nerve impulses, and also in blood coagulation. Calcium is actually factor 4 among the coagulation factors, and it will bind to prothrombin. It will bind to prothrombin. Uh, 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 the gamma carboxy glutamate residues in prothrombin and during thrombin formation. So calcium is also required as a hormone in the certain hormone formation: insulin, parathyroid hormone, calcitonin, and vasopressin. Calcium will also act by activating enzymes, and calmodulin is one of the important calcium binding regulatory proteins. You remember when you talked about calcium handling of the cell, we said that most of the calcium is within the endoplasmic reticulum. It's also bound to mitochondria, and then it's also bound to the calcium binding regulatory proteins, which is calmodulin. So this is the third place where a lot of calcium is bound. Calmodulin has a molecular weight of 17,000 kilodaltons, and it binds to up to 4 molecules of calcium. So once it binds, then it leads to activation of the enzymes, and therefore calmodulin becomes an important regulator kinase. So usually when enzymes are activated by calcium, then they become more effective in the biochemical process. And examples here are pancreatic lipase, enzymes in the coagulation cascade, and also renin. Calcium is also important as a second messenger, just like cyclic AMP for most of the hormones such as epinephrine, especially when you're thinking about glycogenolysis of the liver. Calcium also acts as a third messenger for some of the hormones like vasopressin, where it will act through cyclic AMP. So here, cyclic AMP becomes a second messenger, and calcium then becomes a third messenger. So calcium can be both a second messenger and a third messenger. The myocardium, you find that calcium will prolong systole, and in hypercalcemia, calcium arrest is seen in systole. How do you regulate the plasma calcium levels? Usually we have three main organs: the bone, the kidney, and the intestines, and we have three main hormones: calcitriol, parathyroid hormone, and calcitonin. But we also have other hormones that play a role in regulating calcium, and these include growth hormone, glucocorticoids, estrogens, testosterone, and thyroid hormones. Let's look at calcitriol. The way calcitriol acts on bone is that it stimulates calcium uptake for deposition of calcium phosphate, and it does this by activating the osteoblasts. So therefore, calcitonin is very essential for bone formation, and this is usually vitamin D. And so along with the parathyroid hormone, it increases the mobilization of calcium and phosphate from the bone, and therefore it increases plasma calcium levels and plasma phosphate levels when they are low. It also will act on the kidneys by minimizing the excretion of calcium and phosphate by decreasing their excretion and enhancing their reabsorption. So mainly calcitriol will be trying to conserve and reduce calcium wasting by the kidney. In the intestine, it will increase intestinal absorption of both calcium and phosphate, and also it does this by binding to the cytosolic receptor to form a calcitriol receptor complex. This complex will interact with DNA and to lead to the synthesis of a specific calcium binding protein, and this protein will increase uptake by the intestines. So let's talk about parathyroid hormone. Parathyroid hormone is secreted by two pairs of parathyroid glands, which are usually anatomically located posterior to the thyroid gland. So parathyroid hormones has a molecular weight of 95,000. It's a single chain polypeptide with 84 amino acids. Originally, just like most of the other protein hormones, peptide hormones, it's a pre-pro hormone degraded into a pro hormone and then into an active hormone. So the rate of secretion of parathyroid hormone is usually promoted by low calcium levels. So low calcium levels are the trigger for PTH secretion. How does PTH work? So basically on bone, it will cause decalcification and demineralization of bone, which is a process that is carried out by osteoclasts. And this is brought out by pyrophosphatase and collagenase. These are the two enzymes that are important, and these enzymes will affect bone resorption. So demineralization then will lead to the breakdown of calcium and phosphate that was there in bone into the blood, and this increases the amount of calcium that we have in the blood. Parathyroid hormone will basically act on the kidney by increasing calcium reabsorption in the kidney tubules, and there's a rapid action of elevating blood calcium levels, but it will also promote the production of calcitriol, which is 125 dihydrocholecalciferol in the kidney, and it does this by stimulating 1-hydroxylation of 25 hydroxycholecalciferol. In the intestine, it will increase intestinal absorption of calcium by promoting the synthesis of calcitriol. Let's talk about calcitonin. Calcitonin is a peptide containing 32 amino acids. It is secreted in the parafollicular cells of the thyroid gland. So remember, a parathyroid hormone is in the parathyroid gland, but calcitonin is in the thyroid gland. And the action of calcitonin on calcium is actually the opposite of what a parathyroid hormone does. So there's an element of antagonism, as opposed to the synergy that we were able to see between parathyroid hormone and the calcitriol. So calcitonin will promote calcification by increasing the activity of osteoblasts. It will decrease bone resorption and increase the excretion of calcium in urine. So generally what calcitonin does, it decreases the levels of calcium and the actions of calcium within the blood. Let's talk about phosphate. Phosphate is widely distributed along the body and usually combined with calcium. You remember we said the amount of calcium we have is about 1 to 1.5 kilograms. Again, here you see we have calcium being about phosphate being about 1 kilogram. And 80% of the phosphorus is found in bones and teeth in combination with calcium. And we have 15% of the phosphorus that is usually in the soft tissue. So we know that phosphate is important for ATP or most of the enzymes within the body, both intracellularly and extracellularly, and therefore it becomes very critical to have it also in the soft tissues. It's bound to phospholipids as phospholipids. It's bound to lipids as phospholipids, bound to proteins as phosphoproteins, to nucleic acids, and to nucleoproteins. And that's how it's able to effect its action. Usually, we only have about 1% of phosphorus in the cellular fluid, and most of it, about 5 grams of phosphorus in the brain and 2 grams of phosphorus in the blood. And usually, phosphorus is present both in an organic and inorganic way. Where do we get phosphorus in our food? Usually, we find that foods that are rich in calcium are also rich in phosphorus. So, you're looking at milk, cheese, beans, eggs, cereals, fish, and meat. And milk is a very good source of phosphorus and contains about 100 milligrams per deciliter of phosphorus. So, the daily requirement of phosphorus is about 800 milligrams per day. And remember, we said micronutrients are usually the ones we require by less than 100 milligrams in a day, and macronutrients are the ones we require for more than 100 milligrams in a day. So phosphorus then becomes a macronutrient. So during pregnancy, the level also increases. You can see it's equivalent to the calcium levels that we require. It's 1,200 milligrams per day. What are the functions? Phosphorus is essential for bone formation, bone and teeth formation, and usually in its organic form, it constitutes the hydroxyapatite in the bone, and it provides as an energy source. And we know that high energy is important. High energy is important for ATP, ADP, and all the energy dependent processes. It's also important in the formation of phospholipids, phosphoproteins, nucleic acid, and nucleotides, which are important for most of our biochemical and metabolic actions. Phosphates are usually present in nucleotides, some of which functions as coenzymes. NADP, and flavin coenzymes. And several of these enzymes and phosphates are activated by phosphorylation. There's usually a lot of phosphorylation taking place during metabolism, and this phosphorylation of phosphate. And when you have a mixture of phosphate and monophosphate and diphosphate, you'll end up having a buffer which plays a key role in. You're saying the slides are not clear. So are you able to see them now?
No.
Connection is good. So we have the formation of. Are you able to see?
No.
You're not seeing. And now are you seeing? I think they are blurry because I'm using a PDF. Are you able to read the words?
No.
most of their functions, and the functions that include phosphorylation and dephosphorylation of most of the products. And usually it's formed in as a monophosphate or a diphosphate. It's also important in the creating the phosphate buffer, which has a role in the maintenance of pH in the body fluids. It's also important in the formation of phosphate esters such as glucose 6-phosphate that are important for glucose metabolism and glucose absorption and breakdown. So how is phosphorus absorbed and regulated? Most of the phosphorus comes from the diet. So 90% of the dietary phosphorate is absorbed. Remember, only 40% of the calcium in the diet is absorbed. So we absorb more phosphorus than we absorb more phosphorus than calcium across the gut. So phosphorus is absorbed from the small intestine, and the absorption is stimulated by both PTH and calcitonin. The calcium phosphate ratio in the diet will affect the absorption and the excretion of phosphorus. And you find that when you have the regulation of calcium, are you able to see?
Hapana.
And now are you able to see? Regulated under similar conditions by mechanism in the kidney with respect to PTH and calcitriol. PTH is normally excreted by the kidney, and usually you find that PTH will increase calcium and phosphate levels from the bone and decrease the loss of calcium and increase the loss of phosphate in the urine. So on excretion, 500 milligrams of phosphate is excreted through the kidney every day, through the urine every day, and the phosphate excretion is influenced by many factors including muscle mass, renal function, and age. The renal threshold for phosphorus is about 2 milligrams per deciliter. So if the plasma levels exceed 2 milligrams per deciliter, then you get phosphaturia. And we normally say that phosphate has a PTH has a phosphaturic effect. So phosphates are mainly excreted by the kidneys as sodium diphosphate through the urine. So phosphorus, we find that 90% of the phosphate that is filtered at the glomeruli is reabsorbed by the tubules. PTH will decrease the reabsorption of phosphorus from the proximal as well as the distal tubules and cause increased excretion of phosphorus in urine. Only small amounts are excreted in the feces, and we have a normal range of about 0.9 to 1.5 millimoles per liter in adults, and in children it's 1.45 to 3 millimoles per liter. So in children you have a higher normal range because of their developmental needs. Calcium and phosphorus have a reciprocal relationship. So you find that if phosphate rises, calcium falls. So in the fasting phosphate levels are usually higher. After a meal, we have a decrease in phosphorus, and this, you remember, 90% of the phosphorus is absorbed, and this is due to utilization of phosphorus for metabolism. So the body uses almost all the phosphorus it takes in for metabolism. Red blood cells and white blood cells contain a lot of phosphorus because they require a lot of energy for their function. Hemolysis should be prevented when blood is being taken for phosphate estimation. Otherwise, you'll end up with a higher than normal expected level of phosphate. And that is the end. Does anybody have a question? You are still not able to see. The differences between micronutrients and macronutrients is based on their daily requirements, the required daily allowance. Basically, we said that any nutrient that you require more than 100 milligrams per day is a macronutrient. Is a macronutrient. Any nutrient that will require less than 100 milligrams per day is a micronutrient. And that's the difference. Another question? Metabolism and diabetes. Please let me know whether you can see the slides. Can you be able to see the slides? Can someone respond so that we continue? Are you able to see the slides?
Yes, but they're not yet clear.
And now, are you able to see?
Still not clear.
And now, are you able to see? Is it clearer now? Please let me know so that I can proceed. What is the class email? Just type the class email, then I'll send them to you so that you can be looking at them as I teach. Type for me the class email. you really need to. Diabetes is very common, and you'll be able to see it in your everyday practice. So by the end of this lecture, you should be able to define glucose homeostasis and understand its importance, and also to distinguish between the fasted and the fed state. You should be able to classify diabetes and understand its pathophysiology, and also understand the physiologic principles in the management of diabetic emergencies. And these ones you can get at home, in the hospital, or any other emergency. So the pancreas basically has an exocrine and an endocrine function. Its exocrine function is mainly from the acinar cells by secreting digestive enzymes, and its endocrine function is through the islets of Langerhans, which has the alpha, beta, delta, and pancreatic peptide cells. So we have pancreatic islets here. So have you received? I sent to MBCHB also, underscore. So alpha cells will secrete glucagon, they will raise the blood sugar, and most of them are peripheral. Beta cells will secrete insulin, they will lower the blood sugar, and they're usually central, and they're usually more abundant. We also have the delta cells that secrete somatostatin, which inhibits glucagon, and they are usually more rare. So in glucose homeostasis, we are looking at how serum glucose levels are kept at a steady state, and it's usually achieved by hormonal action. So typically for someone to be considered to be diabetic, they must have abnormal blood sugars. So for someone who is non-diabetic, before a meal, the level should be between 4 to 5.9. This value can vary between the labs. So basically you would need to find out what the normal levels for each lab will be, but generally it's between 4 to 5 mLs per liter. Then after 90 minutes, that's after eating a meal, it should be less than 7.8. For type 2 diabetics, it goes up to 4 to 7 millimoles when they are fasting, and it can be under 8 millimoles per liter after they take a meal. For type 1 diabetics, upon waking up, it's 5 to 7 millimoles per liter. Then before a meal, 4 to 7, after a meal, so how do you interpret this finding that and 2 hours after 75 glucose load, which is the oral glucose tolerance test, then you have a normal result. For someone with impaired glucose tolerance, it means they're not diabetic, but their blood sugar is not normal. It's between 5.6 and 7 millimoles, and between 7 before eating, and 7.8 to 11 after eating. And someone who's diabetic is above 7 and 11.1. What is the importance of this? The brain usually requires glucose as its sole energy source for metabolism, and therefore it's important to ensure that glucose homeostasis is effected. And this is the reason why we have both acute and chronic complications when you have very low blood sugar, or when you have very high blood sugar. And therefore, when it comes to glucose homeostasis, we have two organs that are very key. You remember for calcium, we said we have the intestines, the bone, and the kidney. So for glucose, you have the pancreas and the liver. And this is basically how it's regulated. So you start off with very high glucose. When the glucose is high, then insulin is secreted. Insulin will promote glucose absorption in muscle, in adipose tissue, and in the liver. This will increase glycogenesis and also reduce gluconeogenesis, and this will lower the amount of glucose that you have in the blood because you will have used it in the peripheral tissues. When that is lowered, then you're going to have an imbalance, and therefore you shall excrete glucagon. Glucagon will break down the glycogen that was there in the liver and increase gluconeogenesis. And by doing this, it raises the blood sugar. So you can see you have a nice negative feedback loop within the pancreas and the tissues. So basically, the hormones that are important is insulin and insulin-like factors, and they are also antagonized by insulin antagonists that we shall discuss. Let's talk about insulin. Insulin is a peptide hormone, and it basically has is secreted by the beta pancreatic cells, which are anabolic and are secreted in response to the elevated glucose levels and acts to reduce synthesis or gluconeogenesis, shuttling of glucose into the cells, and lipogenesis. This is the insulin receptor, and basically you can see it's bound to tyrosine kinase, and it's important for glucose transportation, fat synthesis, protein synthesis, and growth hormone expression. These are the insulin-like factors, and several they're called insulin growth factors like somatomidin, and they have a role. Insulin growth factor 1 and 2. They have similar actions of insulin, but weaker than that. Then the insulin antagonists are the ones that oppose the action of insulin. This means that when the blood glucose is low, they'll try and break down glucose from where it has been stored so that the body can be able to increase the glucose levels. Glucagon is top on the list. It is secreted by the alpha pancreatic cells. It's a primary insulin antagonist, and it's important in gluconeogenesis, and it increases glycogenolysis. So glucagon will prevent insulin from forming more glycogen, and also continue the breakdown of glycogen, and also the action of insulin in breaking down amino acids and free fatty acids into glucose. Growth hormone is secreted by the anterior pituitary by the somatotrophs. It increases lipolysis, and therefore it antagonizes insulin. Adrenaline from the adrenal medulla will stimulate gluconeogenesis. It has a flight and fight hormone, and is usually secreted by the adrenal cortex and upregulated in gluconeogenesis. So during the fed state, we have absorption taking place after a meal, and digestion and absorption will also will take place. Glucose levels are elevated, and they will trigger the insulin secretion, resulting in shuttling of glucose into the cells and upregulation of gluconeogenesis. So this is the fed state, where basically you shall see ultimately we have digestion of the nutrients taking place, and the first thing that you basically will see that in the absorption, the liver cells will convert the excessive glucose to glycogen for storage, and amino acids will also be converted into ketone bodies that can later be converted to acetyl-CoA. In muscle, the muscle will convert the glucose to glycogen for storage, and to be used to synthesize actin and myosin. And in the absorptive phase, the adipose cells will store the excessive lipids and increase their fat reserve. In the fasted state, you have the post-absorptive, which is also called the post-absorptive state. This will be geared towards glucose absorption, and it will basically be important for storage. It will occur within the context of extended times between the meals, and it leads to progressive decline in serum glucose levels. This decline will then lead to a decline in insulin levels and their action, and upregulation of glucagon to be able to maintain the steady state. So here we find that in their post-absorptive state, you see you have a lot of very little sugar in the circulation. So what happens here is that the liver breaks down its glycogen to be able to form glucose, and ketone oxidation will also take place to release lipids so that they can also be broken down to form glucose. In muscle, glucose will be released into the blood, and amino acids will be broken down to be able to provide the glucose and also to generate ATP for its functions. In adipose tissue, we find that most of the cells will release the stored lipids and break them down to form glucose. So in the starvation state, we have early and late starvation. Early starvation will occur within 24 to 72 hours after the last meal. They are not moving. Where did I lose you?
From the start.
From the start. Yeah, let me just continue because the time is very short and we just be losing a lot of time. So basically, we are looking at we talked about insulin antagonists. We are looking at the fasted state, what happens in starvation. So we have early starvation and late starvation. So basically, we have early starvation taking place within 24 to 48 to 72 hours after the last meal. And here we find that because the brain specifically depends on glucose as its source of energy, the other peripheral tissues then will begin to switch to fatty acids so that whatever glucose is available is preserved for the brain, since the brain can only use glucose. So we have a glucose fatty acid cycle, which will switch to ensure that free fatty acids are used for fuel. So the dietary fuel is unavailable because somebody is fasting, and the liver glycogen then will remain to maintain the glucose. So you have a lot of glycogenolysis taking place. So there's complete dependence on hepatic gluconeogenesis, primarily lactate and alanine, which we said we break down amino acids. Late during starvation, ketone bodies are used as a major source of fuel for most of the organs, and you find that fatty acids and triglycerol are converted to ketones, and they continue protein breakdown. So if these stores are depleted, the proteins from the muscles are broken down for glucose, and therefore you find that the survival will depend on the amount of glucose, the amount of, 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 of fat. So these are the phases of glucose homeostasis. We have phase 1, phase 2, and phase 3. And here we'll be looking at the nutritional status, the source of glucose, the tissues using glucose, and the major fuel. So in terms of nutritional status, a well-fed state is phase 1, a post-absorptive phase is phase 2, and a fasting phase is 3, so that by the time somebody gets prolonged fasting and starvation, that ends up being phase 4. In terms of sources of glucose, when you're well fed, your source of glucose is diet. In the post-absorptive phase, the body will break down the glycogen and also gluconeogenesis from amino acids and free fatty acids. From in phase 3, when you fast a bit longer, the liver, the kidney will also take part in gluconeogenesis, and this will continue on up to phase 4. In terms of which tissues glucose, but when you get the post-absorptive phase, all tissues will utilize glucose, but will have slow utilization in the liver, the muscle, and the adipose tissue. Remember here, the action of insulin has been reduced, and insulin was a primary mobilizer of glucose within the liver, the muscle, and the adipose tissue. As you fast even longer, you find that cells such as the brain, the red blood cells, and cells that lack mitochondria will actually utilize glucose and small amount of muscle. So in phase 4, due to prolonged starvation, the brain will also utilize glucose at a slow rate, but the red blood cells will utilize at a normal rate. So the major fuel for the brain in the fed state is glucose, and this is maintained in the post-absorptive phase because the body is still absorbing. But once you start fasting, ketone bodies are added as a major source, and this continues on even with prolonged fasting. So let's talk about diabetes. We have a syndrome where you have reduced activity or complete or absence of insulin. And we have type 1 diabetes, type 2 diabetes, and also autoimmune destruction of the beta pancreatic cells. Remember, we said they are central and they are most abundant cells within the islets. So this results in absolute insulin deficiency. And therefore, we find the presentation of the beta cell peptides by antigen presenting cells to the autoreactive CD4 T lymphocytes will lead to the activation of autoreactive CD8 T cells, and this will lyse the beta cells. So we find that then the beta cells are then destroyed by the pro-inflammatory cytokines. In gestational diabetes, we are looking at the degree of glucose intolerance taking place throughout pregnancy, and is normally recognized at the onset of the pregnancy, and the insulin resistance will be worsened by the action of the pregnancy hormones, since they are part of the insulin antagonist hormones. However, the precise mechanism of gestational diabetes is not known. Type 2 diabetes, this one is more common, is because of regular relative insulin deficiency, which is compounded by insulin resistance. So in the pathophysiology, we shall look at the mechanism of insulin resistance. We shall also look at hyperglycemia and its complications and its alternative energy sources and implications. So in terms of mechanism of insulin resistance, here we have an alteration in the insulin signal pathway, and this is due to the mutation of the insulin receptor coupled with a decrease in the number and activity. Then you have coupled with the defects in the function of GLAD4, you have an increase in protein kinase C isoforms that will catalyze the phosphorylation of tyrosine and serine in the insulin receptor. So basically, where we saw the insulin receptor bound tyrosine kinase, we have a change in this activity because we are not able to, the body is not able to express and form GLUT4 on the surface, and therefore the two protein tyrosine phosphatases have been shown to negatively regulate insulin signal, so that protein tyrosine phosphatase 1b and leukocyte antigen related are reported to be elevated in insulin resistant patients. Then the other thing that is important in insulin resistance is inflammation. And here we have altered adipokine secretion and pro-inflammatory cytokines. And here we are looking at tumor necrosis factor alpha, interleukin 6, interleukin 1 beta, which have been shown to decrease GLUT1 and GLUT4. Hyperglycemia is also important. It results from decreased shuttling of glucose into the cells, and the absence of glucose in the cell results in excessive glucose presence within the circulation. So the body compensates by increasing gluconeogenesis, and this compounds the situation even more. And this is why patients will present with symptoms that are related to impaired, increased glucose renal excretion and osmotic diuresis. So a diabetic will present with glycosuria and polyuria. Also, you find that this polyuria, since you're losing, polyuria is defined as when you lose more than 3 liters in 24 hours. So the patient gets dehydrated, and ultimately this leads to polydipsia, that is excessive thirst. Also, we have alternative energy sources in diabetes, where now because glucose is lacking, cannot enter the tissues, the body will break down the proteins and the fatty acids that are available, and this disrupts the osmotic gradient, and it leads to loss of intracellular potassium and water. And this is compounded by excessive amino acid liberation from protein catabolism. So the ketoacidosis that results from fat metabolism is usually one of the factors that leads to acidosis in diabetes, and we know that bicarbonate is an important buffer, but is normally inadequate to be able to prevent this acidosis from forming. Polyphagia, which is what excessive hunger or excessive eating that you get from a patient with diabetes, it's usually as a result of depletion of the energy stores, and usually because the glucose is not entering the tissues. So this is a schematic diagram of what happens in diabetic ketoacidosis based on what we have discussed. So basically, when you're trying to manage diabetes, you want to replace the fluids to be able to restore the volume and also to restore the osmolarity and also to reduce losses. By correcting the electrolytes, majorly you're focusing on replacing, addressing the hypercalcemia, because insulin therapy will shift the potassium into the cell. So whenever you give insulin, you find that it shifts the potassium into the cell. So you have to give insulin and potassium together. And sometimes you find that salbutamol is also used for nebulization for electrolyte imbalance. But it's important to ensure that you monitor the potassium so that hypocalcemia does not result. In terms of hormonal correction, usually insulin is given, and we also have some drugs that are analogs that will allow insulin to be produced from the pancreatic cells if it's available. And incretins will also usually address the serum glucose elevation. So we have amylin and incretin, which will inhibit glucagon secretion, and we have the SGL2 inhibition that will also prevent renal glucose loss. These are the acute and chronic complications of diabetes, and you shall come across them when you're doing your clinical work. We have hypoglycemia, hyperglycemia, diabetes ketoacidosis, HONC, hyperglycemic hyposmolar ketoacidosis. And in terms of chronic disease complications, we have microvascular and macrovascular complications. So microvascular complications will affect the eye, the kidney, and the nerves, while macrovascular complications will affect the heart, the blood vessels, and the brain. This is an example of what happens in diabetes emergencies and just an algorithm for diabetes. And that is the end. Does anybody have a question? Does anybody have a question? Macrovascular, we said, is due to the ones that are related to the heart. Macrovascular complications, we said we have two different types of complications. We have microvascular and macrovascular complications. So macrovascular will affect the heart blood vessels, the coronary heart vessels. It will also affect the peripheral vessels. So that's why somebody will get a heart attack, a myocardial infarction, with a macrovascular complication. Sometimes you find diabetics lose their foot because they don't have blood flowing through the foot, so the foot starts rotting, and that is peripheral arterial disease. And stroke is one of the most common complications of diabetes, which is cerebrovascular disease. So those are all macrovascular complications. Any other question? Yeah, microvascular, you remember we said microvascular is then the opposite. Microvascular is then the opposite. Microvascular is that when you have the small blood vessels going to the retina, and somebody is asking why you get blind. So because the retinal blood vessels are the ones that feed the optic nerve. Once they get blocked because of atherosclerosis and fat deposition, then you actually get blind. That is diabetic retinopathy. Microvascular also can be diabetic nephropathy, where it affects the nerves, and that can also lead to you losing your limb as a result of not having a sensation. Then you also have the neuropathy. Then nephropathy is when you have kidney failure as a result of diabetes, where it affects the blood vessels within the kidneys. So someone is asking, I think I've answered Maxwell, how diabetes affects eyesight. It acts on you have retinopathy, it acts on the blood vessels going to the retina, and therefore, because they are blocked, then the optic nerve does not get glucose, and therefore you end up with blindness. Then Olo is asking, how is dehydration in HONC worse than the one in DKA, yet HONC is in type 2? The main thing is because HONC has a lot of osmotic diuresis, more than a DKA. DKA is more of an acidosis. So you need much more fluid because you have much more glucose. You have higher blood glucose levels in hyperosmolar. You have hyperglycemia, and that's why it has a problem. Sample questions for revision, I think you can check in the library or in the website. So I think we've finished now. If there are no other questions, I'd like to thank you for attending the class. Thank you.